The overall goal of this procedure is to estimate accurate visual population receptive fields. This is accomplished by first preparing effective and efficient visual stimuli localized in the visual space to elicit a distinct and robust response. The second step is to image functional brain activity with high visual attention and minimal movements of eye and head. The final step is to pre-process the acquired data. Ultimately, application of a proposed method to model fMRI signal response to visual stimulus yields the topography of visual population receptive field and its model.
The main advantage of this method over existing methods like direct fitting method is that our method does not require any prior pRF model chosen.
To prepare a stimulus protocol that is effective in eliciting a reliable retinotopic visual response, first, present bar stimulus drifting across the screen sequentially along 8 directions in 45-degree steps. Ensure that the motion is in synchrony with the scanner frame acquisition, so that the bar moves a step once an fMRI frame starts and stays at the new location until the frame ends. To define a field of view in visual angle over which the stimulus is presented, present the moving or flickering checkerboard patterns within the bar to elicit strong visual responses. Then, input the specific parameters such that there are 8 evenly spaced directions of motion, and the bars move by half the bar width per frame. Next, scan the brain of a subject in an MRI scanner using a typical echoplanar imaging scanner.